Welcome to the research talk part of the day. Today is a more evolution and ecology themed day. We have two talks this morning session, one talk in the afternoon. The first talk in the morning session is given by Michael Desai from Harvard. Go ahead.
So I'm going to be talking about inferring structure in high-dimensional genotype-phenotype maps. So before I get going, I just want to make sure I acknowledge folks who actually did the work. So I'm going to talk about experimental data in budding yeast that was collected and through a bunch of really quite impressive experiments by Catherine, Alex, Arthur, Shreyas, Elliot, and then also about analysis that we're doing, a lot of which was done by Gautam, who you all know, as well as Sam Petty, Pankaj, Krishna, and a little bit by Caroline. Okay. So what I'm going to talk about is how we infer genotype-phenotype maps, across the entire genome. And basically, any kind of approach to doing so, basically what people do is you get a collection of individuals with some sort of genetic diversity between them. You sequence them or in some other way quantify this diversity between these different individuals. You measure phenotypes that you're interested in, corresponding to each of those individuals, and you use statistical associations between the genotypes and the phenotypes to try to map the genetic basis of the trait. Okay, so we're going to sort of use these kind of diagrams throughout the talk. So basically the idea is the genotype of each individual is going to be a set of zeros and ones, or minus ones and ones if you prefer, corresponding to the genotype at some set of alleles across the genome that are segregating between the individuals, in other words, at which some of the individuals differ. We've got a bunch of these different genotypes. Each of these genotypes then has a growth rate or some other phenotype in each of some set of environments, which we'll denote by alpha, okay? So the problem is high dimensional typically in that there's a large number of loci, so there's some enormous high dimensional genotype space. And we'll also be talking about high-dimensional phenotypic spaces. So we've got phenotypes of each of these genotypes in a bunch of different environmental conditions. So that'll be high-dimensional as well. Okay. So there's whole different fields in biology, quantitative genetics, classical genetics, that are associated with trying to solve this problem. So all of classical genetics is basically about making mutants, sort of making variants and measuring corresponding phenotypes and working out the structure of genetic pathways that way. Nowadays, we have more systematic high throughput methods like CRISPR of making individual mutations in a whole bunch of different genetic backgrounds of lots of different kinds of organisms, including in human cell lines, measuring phenotypes. And because you're making single mutations at a time, you can more unambiguously say what each of those mutations does. But at the same time, you have practical limitations on how many different things you can screen, problems with, so basically there's sort of low range of different diversity, and you can't identify interactions between different mutations. You're sort of looking at effects of single mutations on a single focal genetic background. Okay. In organisms like humans, of course, we can't do a lot of these kind of manipulations, although we can in human cell lines, but not in whole organisms. So instead, we're restricted to looking at genetic diversity as it exists in nature. So we can take a bunch of individuals, we can sequence their genomes, we can measure phenotypes, and now there are huge, biobanks of this kind of information approaching millions of individuals, like the UK Biobank or the All of Us Biobank in the US. And this is called GWAS. And I'm not going to talk about this much today, although maybe if I get far enough at the end, I'll talk a little bit about work we're doing with these GWAS systems. What I'm going to talk about is a third kind of approach, which involves engineering of diverse populations in organisms where we have control over, the control over mating, essentially, that we can construct populations how we like. So this includes, model organisms like yeast or mice or Drosophila, as well as, of course, crops like corn or wheat or whatever. People do this. So basically what you can do is you can take parental strains, you can mate them in various ways as you see fit, and then you can construct individuals that differ genetically according to the way that meiosis works, okay? So I'm going to talk about data that my lab generated using this kind of approach in budding yeast, but it's the same idea in really kind of any model organism. We take two parental strains of yeast. I'll talk about what these are in a minute, but they're two different haploid parental strains. So they'll have what we'll call the blue and the red genome. You mate them, you create a heterozygous diploid, you sporulate that diploid to get haploids. Each of these haploids has a kind of genome that's a mosaic of the parental genomes. So typically there's about 60 breakpoints per yeast meiosis per generation. Yeast actually has a fairly high mutation rate relative to the, recombination rate relative to the genome size, which is one advantage. So there are about 60 breakpoints. So there'll be like about 60-ish patches of red and blue, okay, across the genotype. We array each of these individuals in these microplates. We sequence them, I'll talk about that in a second, and then we measure phenotypes, okay? So this is classically how people do this kind of a thing. And then the sort of classic approach to mapping the genotype-phenotype map is basically doing doing linear regression, doing a series of kind of tests where you sort of scan across the genome. At each locus in the genome, you look at the phenotype of all the individuals that have the blue allele at that location and the phenotype of all the individuals that have the red allele at that location. And so you get some cloud of phenotypes of individuals that have blue and have red, and then you do some kind of statistical test to ask whether these distributions are different, and you compute a p-value from that. Okay, so in this example, at this location, there's a difference, at this location, there's not. Sorry, go ahead. Yeah. Right. For each segregant, we call these segregants or offspring of the cross. There's random breakpoints. There's whole stories that you can tell about what the recombination map is. It's not like a uniform distribution of breakpoints, but it is random in each individual. Yeah. So they differ in the cross that I'm going to talk about at 42,000 SNPs across the genome. Okay, so basically what you would say then, okay, if there weren't this recombination structure, what you would say is that this locus causes if you have the red allele at this locus, your phenotype is greater than if you have the blue allele. And the difference between these averages would be like your estimate of the effect size. Okay. But of course you have a problem that because there's only 60 breakpoints and 42,000 SNPs, nearby SNPs are likely to be highly correlated. If you have the red allele here, you have the red allele very likely nearby until you get to one of those breakpoints. So what that creates is sort of phenotypic associations that aren't causal. If there's some causal locus nearby this locus, you'll get a phenotypic association, okay? So that's going to be a big problem, of course. But what we do then, what people do traditionally is you create some LOD score, some log odds ratio of a difference as a function of genome position. You plot that and you see some result like this, okay? And you sort of would infer here, there's some causal locus somewhere here. There's some peak of association there. It sort of declines as you go away from the causal locus because you're getting less and less linked to the causal locus. You're more and more likely to have had a recombination event. And you can see, okay, here there's there's some overall genome-wide significance threshold, and you have some dozen or so causal loci, and then association via linkage. Okay.
Michael?
Yeah.
Just to follow, so here also in this plot, these light and gray sort of demarcate the breakpoints.
These are, no. Each individual in the cross will have different breakpoints. These gray lines, there's 16 chromosomes in yeast. So there's always obviously a breakpoint between chromosomes, right? The chromosomes do segregate essentially independently, hopefully, but within the chromosomes, there's linkage. Okay. All right. Yeah. We're going to get to that. Okay, but this figure, okay, is from old collaborators of ours, okay, from some other work that they did. The Krugliak lab is sort of, Leonid Krugliak's lab at UCLA is sort of done many of these kinds of things. What they do is they array these things on agar plates, 384 or even 1536 well plates, and they measure like a colony size as a sort of proxy for growth rate. We're going to talk about sort of a little bit more modern methods of doing that in our data. But that's, and so then they'll have a bunch of different phenotypes that correspond to different media conditions, different drugs they put on the plate or whatever it is. Okay? All right. So in these kind of QTL mapping experiments, typically I mean, big agriculture companies do bigger things, but in most model organisms, you get up to at most about 1,000 organisms, 1,000 sort of segregants in the cross, right? Obviously, the power that you have, sorry to go back here, to distinguish these things depends on how many dots you've got, right? You've got 1 dot for every segregant, and the more dots you've got, the more finely you can resolve differences, okay? And so typically people have done up to 1,000 segregants. Obviously, this is most in an organism like yeast, less in Drosophila, even less in mice, okay? People have worked to improve this by doing deeper crosses. So we took 2 parents, we mated them, we created children. You could mate the children to create grandchildren or great-grandchildren or great-grandchildren. And every time you do that, you create more breakpoints. So you reduce this association problem. You cause other problems, which I'm going to talk about in a minute, but you reduce this linkage problem, okay? And you can more finely map what the causal loci are. You can also survey diversity more broadly. So of course, you're limited to looking at the effects of mutations that differ between the 2 parents. So you can get more parents and do various kind of circular crosses or common garden things or different stuff like that. Or you can just do more. Okay, and so my lab, we set out to just do more, okay? Because, you know, you can't do this or this in humans, but this you can do, okay? We have, you know, we've got a couple million, we can get up to like 7 or 8 billion, you know, that's gonna be the limit. Okay, so this is work by these folks, okay? And we set out to increase the scale of these things by 2 orders of magnitude. Okay, just to sort of see what one would get when one started doing that kind of stuff. And it also enables new kind of statistical approaches to look at, you know, to start grappling with the high dimensionality of the problem, as I'll talk about. All right, I'm not going to talk in great detail about experimental details. I'm just going to run quickly through this. Those of you who are interested, I'm happy to talk about it afterwards, but I want to talk more about the statistics. So I'm going to run through this quickly, but basically, we took these 2 strains. I talked about this is the RM and this is BY here out of the phylogeny of yeast. These are 2 standard laboratory strains, differ, as I said, at 42,000 loci. We barcoded the parents. Okay, so you create like a neutral DNA barcode at some kind of locus that doesn't affect any of the phenotypes that you're interested in. It's highly diverse. Okay, so you basically create a bunch of parents that are identical, except they have a different barcode, thousands of them. You mate them, you create the diploid, you do this funky recombination thing to combine the barcodes. So then you go from like thousands to thousands squared unique barcodes. Okay, so you get millions and millions of unique barcodes in these diploids. Then you sporulate them, and just by chance, every offspring is going to get a unique barcode. So now you've got these diploid strain collections, which I'm not going to talk about, but we have these barcoded haploid strain collections where each offspring has a unique barcode. How we do that is some magic that Alex developed, okay? Then we have to sequence them all. The key sort of insight here is that sequencing 100,000 yeast would be really expensive if you didn't do 2 things. A, like do really cheap library preparations, and B, sequence super efficiently. The key thing is here, because you know what the parents' genotypes are, and there's only 60 breakpoints, you can sequence at much less than 1x and still almost exactly know the genotype of the offspring. So here, for example, is an example of chromosome 13 from one of these 100,000 offspring that we made. And this is with 15x coverage, the genotype, right? So basically what it's saying is that these loci, it's RM, that's one of the parents, then there was a recombination breakpoint, now it's BY, another recombination breakpoint, so on like this. When you do 0.1x, okay, we just do some hidden Markov model to figure out the gene, our posterior probability of being one parent versus the other as a function of genome position. And you can see it almost exactly matches, except there's a little issues right at the boundaries. Okay, these issues at the boundaries are important for fine mapping, basically for distinguishing what is the precise causal locus. But I'm not going to get into that really much today. But And you can quantify the error. Basically, the bottom line is even at 0.1x, you have error rates of less of, miscalling the genotype of, 10 to the minus 4 or less. Okay? Because you do low coverage sequencing, you don't get the barcodes typically. In the sequencing, you have to figure out which barcode corresponds to which genotype. You do that via some funky pooling things. Basically, you make orthogonal pools of offspring. You sequence the barcode in each pool and you look for overlaps. Okay? There's a whole cool problem associated with that. But the bottom line is, at the end of the day, we have the genotypes and the barcodes associated with each of 100,000 offspring. And then we can measure phenotypes. And because each of these things is barcoded, that's what I'm saying here. We do a funky scheme to get the barcode, all right? I'm gonna leave it at funky scheme, okay, because I don't wanna spend 10 minutes, but I'm happy to talk afterwards. Okay, so then we pool these things. We just mix them all together in a flask, which has some media with some conditions, okay? And then we propagate that for a few days. At each day, we take out a small sample, okay? We extract just the barcode locus and we sequence just the barcode locus. And since we're sequencing just the barcode locus, we can sequence it at like 100 million x depth, okay? And from that, we can quantify the relative frequency of each of the 100,000 offspring, and we can see how those change over time, okay? And you get data that kind of looks like this. Each line is the frequency of one barcode on a log scale as a function of time. And you can see the genotypes that kind of grow better in this condition, they go up. The genotypes that grow worse go down. You can quantify that slope, and that's your phenotype, your relative growth rate of that genotype in that media. And you can do this for a bunch of media. The advantage of this is you can measure 100,000 phenotypes in parallel, quickly, efficiently, accurately. This is a typical thing we get, okay? So this is the phenotype of the RM parent, this is the phenotype of the BY parent, and the offspring follow some basically normal distribution, more or less in between the parents, as you would kind of expect if there were like a large number of loci controlling this phenotype and the offspring are kind of randomly getting them. So you get some central limit theorem giving you this. Yeah.
Just to get a sense of this, so the little bit I'm aware of this, usually when these fitness measurements are done, you do it in some sort of condition where there's 95% parent and 5% the rest. Is this something like that?
No, this is 1/100,000th of 100,000 things.
Okay, great.
And so you're effectively measuring the relative growth rate of each thing relative to the average.
Right.
Okay, and there's an inference problem there that I'm not going to talk about, but yeah.
Okay, but basically the parent is is the same as everyone else.
You can think of this, the relative measurements, the relative values matter, but the zero is arbitrary. Okay, and whatever, you can get pretty good correlations here. So the accuracy of this is basically set by the sequencing depth. You could do better if you wanted to spend more money. This is how good we did. All right. You can measure other types of phenotypes as well, okay, this way. Like, for example, you could measure mutation rates this way by like asking, how frequently different genotypes generate mutants that you can select in some way or other sorts of things. Okay, but I'm going to just talk about growth rate measurements today. All right. So the first thing that happens when you do this, okay, is that you immediately see that this traditional method of identifying, we call them QTLs, causal loci. Or QTL is a region of the genome that contains a causal locus that affects the phenotype. Okay? And the traditional method of finding them is just, we plot these LOD scores, testing for association, as a function of position along the genome. And I've just plotted here, subsampling 1,000 segregants, that's the green line here. And if we subsample 10,000, we get this red line. 92,000, which when I made this was all we had clean data from. It looks like this, and here's a few zoom-ins. And what you can see now is that you've got like whole regions of chromosomes that are associated with with this phenotype. And this is generically true, and it's sort of what you would expect when you have linkage, and as you increase the power, the whole genome is linked to something that affects the phenotype. And so it really becomes difficult. Like if I look at this, is this one causal locus here and noise? Or is this 1, 2, 3, 4, 5 causal loci, or is it 1, 2, 3, 4, 5? You know, what's actually going on here? So we need to do something more sophisticated with inference. So the standard way that people do this in quantitative genetics is, this is going back to this sort of idea here. Yeah, sorry. Well, okay, what do I want to, what this is saying, it's not an inference at all, is the point. It's not that it's a bad inference. It's not an inference at all. This is telling you that the whole damn genome is is statistically associated with the trait, which is really an artifact of the fact that your F1s are highly correlated, right? They're linked. If you want to then now, there's sort of two things you might imagine you want to do. I'm going to give you a new genotype that I haven't seen before, predict me the phenotype. Okay, I need to have a model of what the causal loci are to do that. Or the second sort of thing is, I might want to say, all right, I want to make a bunch of drug targets. I want to know what the top causal loci are, you know, and I want to treat those as drug targets for whatever, you know, if the phenotype is some disease, I want to, I mean, obviously that's not relevant for use, but to do that, I need to have some model of what the genotype-phenotype map actually is. Okay. So the standard way of doing this, all right, is you write some kind of expansion like this, okay? The genotype, the predicted phenotype, okay, is some constant plus some additive effect of some set of causal loci, okay? Sum over those additive effects, plus pairwise interactions, plus three-way interactions, plus so on. If you keep all orders of this, it's completely general. But of course, there are 42,000 loci, so there's 2 to the 42,000 beta terms here, okay? So the typical thing is we just truncate this, okay? We'll typically just truncate all these things to 0, okay? And we'll do some best fit here. That's still 42,000 betas, okay? Which is approaching the 100,000 data points that we have. So even that, we need some kind of regularization. And if we want to get pairwise things, we got to regularize the hell out of it, you know, and so on. Okay. But that's what we do, okay? Because I mean, what the hell else are you going to do? Okay, I'll tell you about one other answer to that question that Gautam has been helping us with, if we have time, but that's what we do, all right? And so what you get is something like this. Here's an example, all right? We get, this was that LOD score curve, this t-test thing in this line, and here you can see you identify a bunch of specific loci. Yeah.
So just to check this sort of regularization, is this a sparse L1 one or is the L2?
Okay, well,
does it matter? It depends on whether it is assuming things are sparse or dense.
So
I don't know if you want to get into this.
I mean, I will get into this in some specific examples. You know, I think typically you in some sense should do an L0, but people typically do L1 or L2 because it's computationally more tractable. This is done with some kind of forward search that actually is an L0, but that has other drawbacks. But it's a complicated problem for which there's not really a great answer. Yeah. Okay, but this is what we get with our sort of analysis. You've got a bunch of these causal loci that we identify as passing this regularization. There's some confidence intervals around them. So like this one is low confidence exactly what the causal locus is. This is more confident. In general, the stronger the effect, the more confident you are about the locus, about where it is, where the causal locus is. And so we did this in some initial paper that is now published a few years ago with 18 different environmental conditions. Okay, these are the 18 conditions. These are like this is a temperature gradient in rich media. This is a bunch of other laboratory media. And what this is showing is each of these lines is the sort of the set of causal loci with a color scheme for their effect size. Okay, and so the bottom line is you find some kind of genotype-phenotype map here. Okay, and we can also then start to map epistatic interactions between loci. I'll talk more about that a little bit later. But you get also some interactions, you know, pairwise epistatic interactions between loci. We see some effect size distribution. So there's a small number of loci These are 18 lines, one for each of the 18 conditions. There's some small number of loci where that have a large effect on the corresponding phenotype, more and more that have a smaller effect, and then you get some decay over here due to power. Basically, if things have too small of an effect, you can't detect them. So you find, you know, with 1,000 segregants in earlier work, about a dozen causal loci that affect each phenotype. As we go to 100,000, you increase that to like 100 or a couple hundred. This sort of distribution suggests that if we had more power, we would just get more and more, you know, as we continue to go down. Yeah. So we look for epistasis here, okay? We attempted to look for like whether there's a statistical tendency for like the RM and BY pairs to be higher than the cross pairs. The answer is maybe a little bit, but not really convincing. So the first order answer to your question is no. We looked for that, we didn't see it. Okay. All right. So that's the basic sort of framework of the data. And I guess what I'm going to talk about, depending on how quickly I go, is two sorts of ways of looking, two sorts of new ways, statistical ways of looking at these high dimensional data sets. So the first is going to be, this is all work done with Gautam and some of it with Pankaj Mehta and Sam Petty and others who I'll highlight as we go through. The first is sort of attempting to find lower dimensional latent structure in this high dimensional genotype-phenotype map. Okay? And the second is using attention models basically to try to find an alternative way of capturing the architecture of this map. Okay? So the first, we're going to think about high dimensionality in terms of the number of environments, right? So we can see we've independently inferred these genotype-phenotype maps in each of these 18 environments. But if you squint at this, you can see the map we infer at 23 degrees is very similar to 25 degrees. And 25 is kind of similar to 27. And 23 is similar to 27, but less so than it is to 25, and so on. Kind of as you would expect. You know, suggesting that there's like some kind of underlying phenotype that might be heat tolerance or cold tolerance, and that all these sort of phenotypes that we're measuring are somehow, there's a lower dimensional structure that's like heat tolerance or cold tolerance. And for some of these other conditions, we don't necessarily have an a priori expectation like that, but one might imagine that the structure of genetic organization in yeast is such that there are like various modules and pathways, you know, and you know, there's something like stress response. And, you know, I mean, this is sort of along the lines of what Sid was talking about earlier. You know, there's things that the yeast are doing and there's things we're measuring, and we're measuring things that are linear combinations of the things the yeast is doing. Maybe not linear, but they're combinations of the things the yeast is doing. And what we'd like to kind of find is that lower dimensional space of the things that the yeast are actually doing, right? That's a very crude way of putting it. Yeah. Hmm? No, sorry, these are percent. They differ by 20%, typically, yeah.
It's just like you were talking about temperatures is the obvious thing. Some of the other environments were different nutrients, right? So is there a sense?
Nutrients, different stresses. So this is copper, this is SDS, which is like a soap.
Right.
Raffinose, mannose are different nutrient sources. Ethanol is ethanol, which like yeast has a very special relationship with. Many of you might enjoy that. YNB is sort of a more limited, less rich media. 4-NQO is a DNA damage agent that lots of people have looked at. Soloctidil, this is molasses. So we couldn't do anything more with molasses because the way we did this is Catherine, you know, went to Whole Foods and bought a jar of molasses, and like when it ran out, that was that. It's, you know, no two jars are the same. This is lithium acetate. It's like a metal. Yeah,
I see, because my question was going to be just like there's these different temperatures, maybe there's a sense in which two different nutrient sources are similar enough, and maybe there's a
We're going to get into this kind of thing, but yeah, that's exactly the idea. Okay, so the idea is like along this lines, there's some set of things that yeast are doing. We'll call them core processes. This is a hypothesis, right? And that somehow the idea is there's some genotype which affects the core processes, and then these core processes affect the phenotypes we measure. If we could just measure these core processes, that would be great, but we don't know what they are. We don't know how to measure them. So we measure these phenotypes, whatever the hell they happen to be, and whatever's easy to measure and practical in the lab, and that ideally that we think that varies across these core processes in a way that will allow us to infer them. But then the idea is, okay, how do we sort of find this lower dimensional space of core processes from the data, right? So, all right. The idea that Sam and Gautam came up with, at least as a first pass, which was published a couple of years ago, at least this first version of it, is the idea is essentially this. We have some loci. Each of them affect the core processes via one of the kind of maps that we were talking about before. And then these K core processes each, again, have some map to the E measured phenotypes. Okay? So formally, if we only keep those first order terms, this is just a matrix decomposition problem. That's it. Oh, sorry, go ahead. Yeah. Ideally, yes, but we actually like, the epistasis we have limited power at, right? So we're trying to use just the beta i's to do that. So there's no matrix there. In practice, like we can only measure beta ij's that have a non-zero beta I and beta j, because otherwise you have a 42,000 squared problem. And so one of the things we're trying to do going forward, which I'll talk about, is like exploiting the beta ij's a bit. But for now, like we're just looking at beta i's, and so we can't really do what you're suggesting with just the beta i's. Okay. Well, other people, I'll talk about that a little bit more in a sec. All right. All right. But for now, okay, we just have these additive effects. It's just a linear decomposition problem. We have basically each locus has some effect on process K for the K processes. Then there's this matrix W, which is the sort of map from core processes to actual phenotypes. And this sort of gives us, right, there are E phenotypes and L loci. So there are E by L different values here. There are E by K and K by L values here. So as long as K is small compared to E and L, this is a much simpler description. Okay, many less parameters. All right, so that's what we want to do. All right, the problem is that this matrix decomposition is not unique. Okay, so for example, if you have some sparse generative core process here, so locus, this is just a simplified toy example. Locus 1 affects module, we'll call it sometimes modules. It's synonymous with core processes. Locus 1 affects module 1, locus 2 and 3 affect module 2, and then these affect the things we measure. Okay, you can think of this from like a linear algebra point of view of the measured phenotypes lie on a sort of two-dimensional subspace of this plane. This L to M map are like the basis vectors in this plane, but you could like rotate these basis vectors and you would get an equal goodness of fit, right? So a standard thing people do is like SVD, for example, and they basically choose the basis where, you know, in order each basis vector kind of explains the maximal amount of variance in the measured phenotypes, okay? So that's what, you know, other folks have done things like this. What Sam and Gautam suggested was that, okay, actually, I should just like say, right, the key sort of assumption here, like if these core processes really are like modules or things like that, the idea is the map should be sparse, at least at this level, right? Like there's, each core process involves a relatively small number of loci, and each locus participates in a small number of core processes. Basically, the idea is like each gene does something, and that something that it does is part of some process in the cell. right? And it's not like each locus contributes to all the core processes. No, like the locus is either involved in ribosomes or cell wall or whatever, some specific thing, right? So that's the idea, okay? And so we want to look for like a sparse decomposition, okay? And so we do so basically by imposing a regularization that involves sparsity, okay? And so that regularization essentially breaks this symmetry, okay? Of course, so we want to basically, okay, I don't want to get into the details of like how one does this, but basically we do some decomposition which minimizes some error subject to some sparsity penalty. By definition, that's going to do worse than SVD because SVD just is minimizing this thing and choosing the basis, you know, that just ordering things by the variance explained. Okay, when we impose this sparsity, we will do worse. Okay. But what we can see is that if you simulate a sparse generative core process, okay, then you do SVD or you do this sparse decomposition. If the generative core process is sparse, okay, you will get a low error sparse solution that has almost as low error as SVD, but this will recapitulate the true generative process, whereas this typically won't. Okay, yeah.
Data that you guys input?
Yes.
Okay. Data.
Yeah, exactly. All right.
And Michael, just to get a sense, can you say something about how you choose the number of core processes? You just choose them out of, you choose them arbitrarily until you.
Yeah, I think I say this, yeah, here. We input an upper bound, the regularization typically will not use all of them. Okay, so effectively you're setting that by these lambdas. Okay, and of course, the bigger you set the lambdas, the worse that this will do relative to SVD. So you can sort of, as you tune up the lambdas, increase the sparsity, the error gets bigger, okay? And so you've got some kind of a choice of what you want to do here, okay? So one can do this SSD across a range of these parameters, and then the question is sort of what do you pick, right? And the idea here that was this Sam or you, Gautam, that came up with this? One of you guys, or maybe jointly, it certainly didn't come from me, all right? Had this idea of how to sort of guide this choice, okay? Intuitively, like this idea of sparsity makes sense, but like, is it reasonable, okay, for a specific data set? The idea that they came up with is, if there's a decomposition of this multi-environment genotype-phenotype map that has a sparse, you know, generative process, okay, these phenotype vectors, okay, the actual, you can think of each genotype as having an e-dimensional phenotype, right? And we can think of that as a point in this e-dimensional space, okay? And it will sort of lie on a plane. These points will lie on a plane that can be generated by the sparse vector. So SSD can find this low error solution. Now, if I randomly rotate these phenotypes in this e-dimensional space, okay, just randomly rotate them, okay, it's a mathematical property that they'll still lie on the same low dimensional plane, okay? Rotating them doesn't change the dimensionality that these points span, right? So they'll still lie on a low dimensional subspace, but they'll lie on a sort of random low dimensional subspace, and random low dimensional subspaces typically cannot be generated by sparse basis vectors, okay? So the idea is you take the actual data and you do this sparse decomposition, and then you randomly rotate the data and you attempt to do the sparse decomposition. Okay, SVD will not care about this rotation because it still lies on the same low dimensional subspace. SVD will not see that at all. Okay, but the sparse decomposition should do much worse on the rotated data than the real data if the real data is in fact generated by a sparse process. Okay, yeah. It depends on there being core processes. There's a smaller number of core processes than things we measure. So probably are, right? But the thing is, like, core processes that then don't differ in their effect on those two things, we're going to be blind to them, which is both a feature and a bug. And so let's say that we've got, as we do, 7 or 8 different temperatures, okay? There's a whole bunch of core processes that affect things, but that don't affect things differently across those different temperatures. We're going to be blind to them. There might be a core process, so that's temperature tolerance, and that will affect, systematically across these things. And so that process we can get at with these measurements. We need like a set of phenotype measurements where the core process affects them differently, and there's enough measurements relative to the core processes, right? So the more phenotypes that you have, the better. But to some extent, if you don't pick phenotypes that are differentially affected by a given core process, you'll be blind to that process. Okay. Yeah. It's growth in a variety of conditions. Yeah. You could do other things, but we haven't. Yeah. Yeah. Okay. So the idea here is, like, we can look for this sparsity in this empirical, purely empirical way. Okay, you can do this with simulated data, and you find indeed that, like, as you increase, there's 2 levels of potential sparsity, right? At each level of that map. And what you do find in simulated data, if you simulate sparse data, the sparse solution of the real data is much better than the sparse solution for randomly rotated data, okay? And you can do that for either type of sparsity, okay? And in the real data, this is what it looks like, okay? So you see that indeed there's evidence for sparsity. So you have, the real data, the sparse solution is much better than in the rotated data. And then basically what you can do, this is as a function of how sparse it is. So you can kind of, I don't think we have a good way of really choosing the sparsity from this, but you can sort of see, like, here, like, you probably want to choose something like around here, I don't know, as a reasonable thing, right? And so then you can actually look at what these things are. Okay, so in this case, okay, we have these different environments. These are like representations of those W and M vectors. So this is basically like how each environment depends on each of 8 inferred core processes. And then here's how each locus affects the different core processes. And so for those of you, including me, who have limited knowledge of yeast physiology, this may not mean much. But there is, like, at least some sense in these, although not entirely. Like, so for example, this is a lithium transporter. Okay, this is a ENA1 and ENA5. These are components of a lithium transporter. They form a core process that's important, core process 2, which is important particularly in lithium. Okay, and there are other things as well. All right. I'm going to run quickly through this because I want to at least briefly talk about these attention models. But basically, as Kabir was highlighting, our power with this method also depends on what environments we choose to measure. So we've sort of embarked on a much larger kind of survey of different phenotypes that involve, like, a structured survey. So we have different sort of hub phenotypes, hub environments, and then we apply different degrees of different perturbations. So like different concentrations of cyclohexamide or different concentrations of caffeine in attempt to sort of probe this space a little better, okay? And you get stuff out of this, then you can get more of this kind of stuff, all right? Conceptually, it's the same as what I've been talking about, but hopefully we have more power with more phenotypes, higher dimensional. Okay. We can also measure epistasis. To get to the question earlier about that, I've talked so far just about inferences from these beta terms. We can try to infer something about these beta ij terms, right? That gives you these epistasis maps like this. And traditionally, like, what people have done is attempt to figure out the structure of these kinds of core processes, often with more levels of hierarchy, not just 2 levels, from this kind of epistasis data, looking for structure in this beta matrix, essentially, okay? And the type of data that people use for that is typically like double deletion data in yeast. So folks have made all possible pairwise gene knockouts in yeast and measured sets of phenotypes, and then they look for structure in those matrices, okay? And you can do now, I mean, I've been to this lab, Charlie Boone's lab in Toronto. You've probably been there. I mean, it is amazing. The number of robots they got, I mean, so they're doing all triple deletions now in yeast. So it's like 4,000 cubed genotypes and measuring all these things and trying to get at it that way, okay? And you produce these beautiful things where you've got this hierarchical, they have complexes and networks that then cluster into bio and so on in these matrices. So you can try to do that. And you know, a question is like, that they've just done basically based on certain types of clustering in these matrices. And a thing that we're sort of working on now, but I don't have any results to share with you, but is to essentially try to incorporate epistasis in these maps. And you can get sort of different types of clustering. You could imagine having epistasis at this level and epistasis at this level, and try to infer, use that epistasis to better infer what these core processes are. And that's one thing we're trying to do, right? But I have 10 minutes left or 9 minutes left. And so I want to talk about briefly in that 9 minutes a totally different sort of thing, which is basically now not looking at using the high dimensionality of the phenotype space, but a different sort of way of grappling with the high dimensionality of the genotype space. So we talked a bunch about this classical inference approach, okay? And the unfortunate fact is you've got to truncate this, okay? And so what that means is that sort of fundamentally, like, if you've got, like, say, some higher order interactions, some 4th or 5th order, whatever, interactions among multiple loci that affect phenotypes, what you're going to do instead is you're going to sort of capture those effects incorrectly in biases in the lower order terms, okay? And that may still help you identify the causal loci involved in those interactions, but it won't capture the interactions, okay? And that's potentially a big problem for all sorts of things. Like, one of the potential really sort of practical problems of this in human genetics is that we've got these huge biobanks, okay? They tend to be built and constructed by rich Western nations. So like there's the UK Biobank. We've got a huge amount of data about British people, their genotypes and their phenotypes. If you're British, this is great. But if there's some high order epistasis, you know, if you're Indian or Japanese or whatever, typically the inferences drawn from the British people are much worse in predicting phenotypes in non-British people. There's a variety of potential reasons for this. One of them is these high order epistatic interactions, okay? So how could we potentially capture them? A possibility is to use these attention models that are used in natural language processing. So they're the basis of things like ChatGPT, and which you all are presumably familiar with. The basic idea behind these is to sort of each locus learns through this attention mechanism what loci to pay attention to and what loci not to, and it can be sort of asymmetrical. And you know, the question we had is, with these 100,000 yeast data points, do we have enough data to train these attention models? And if we do, what are they saying about the architecture? Could they potentially capture higher order interactions? Okay. Now, I spend not nearly enough time doing the computation and the theory, and too much time, thinking about the experiments. And so I don't know a ton about this. Gautam and Pankaj know much more, and Krishna, who led this work, but I will do my best to very briefly summarize the basic idea. Basically, what you do is you take the genotypes, you create this embedding matrix, and you basically, each locus in each individual becomes some d-dimensional little vector. And these d-dimensions are going to be like different ways in which that locus can pay attention to other loci, okay? you pass each of these things through a variety of attention layers, and you get kind of a Z prime, Z double prime, and so on. And then at the end, you do linear regression on the Z double primes or triple primes or whatever. These attention layers, basically what they're doing is they're taking each component of each Z, and you learn these query, key, and what's this value vectors, value matrices that basically tell you how each locus pays attention to or is paid attention to by other loci. And then you kind of, you multiply these query and key things together to modify your Z, okay? And it's basically like adjusting each of these genotype vectors according to what the presence or absence of other loci, of other alleles at other loci, okay? And then you do the linear regression at the end on the output of these things, okay? There's all kind of details in this and choices about the architecture that Gautam can tell you all about how we did it wrong, okay? Or how we could do it better. But the bottom line is we tested this on synthetic data where we basically, created a synthetic genotype and phenotypes. What we did was we used the actual yeast genotypes. So we maintained the linkage structure of the data and all that, but we just sort of took the actual genotypes and synthetically assigned phenotypes according to some map that we chose, some genotype-phenotype map that we chose, which basically has some linear terms, and just for the sake of argument, we put in fourth order epistatic terms, and the relative balance of these things is controlled by some parameter epsilon. So we did this the wrong way around. I don't know why. Epsilon of 1 involves all linear. Epsilon going to 0 involves all epistatic. Okay? It should be the other way. But the bottom line is, okay, as you might expect, all the models do really well when the true map is linear. Okay? As the true map becomes more epistatic, okay, the traditional linear or the linear plus pairwise epistasis, the traditional methods get worse. These attention models do pretty well. So they can capture this fourth order epistasis. This does depend on how many causal loci you have. With 100,000 genotypes, we have enough power to train these attention models, if there are 100 causal loci, but not if there are 300. So there's, it's not that they do worse than the traditional methods, but they don't do better once you have too many causal loci relative to the number of genotypes you have. So there is some limit. Okay, they do, okay, this is saying about how they do better. The funny thing is, well, I don't know about funny, it's kind of curious. They don't actually do better on the real data, okay? They do about the same. So, and they do pretty well, okay? So it suggests, at least to me, that we're somewhere here, okay? The real data actually maybe doesn't have that much high order epistasis. So, we do about as well with the attention models as we do with the standard approaches, which I found a little bit surprising. And, we do reasonably well, not fabulous, but it sort of suggests that the gap between how well we do and, perfection is more about measurement errors and other things than high order epistasis. And then the other thing you can do with these attention models is you can sort of jointly train them on all the environments. So we can do something akin to this kind of decomposition of, into core processes. I mean, not in the same way as was done before, but maybe the attention model can sort of implicitly infer core processes across all the different environments. We just include the environment essentially as another component of the input here, okay? And again, what we get is that we do about as well as the traditional methods, but not better. The thing that you can do much better with these attention models here is transfer learning, okay? So imagine that I take away some of the data. So let's say that with the temperature gradient, for example, I've got 100,000 measurements for, a bunch of it, or it doesn't even need to be the gradient, it can be any of these things. Let's say I've got 100,000 measurements for 17 out of the 18 environments, and then I just give you 10 data points for the last one. these attention models can essentially use those 10 data points to figure out this environment is kind of like this one or that one or some combination, and actually do quite well even when trained on only those 10 data points. But the traditional methods can't really do it all. And so this is just showing that, like, essentially, as you reduce the number of data points from 10 to the 5 down to a few for one out of the environments, you still do really well with the attention models, even though you can't do well with traditional methods. Yeah. Yeah. Well, the attention models, they don't know anything necessarily about core processes. They're somehow encoding the relationships between environments in this attention mechanism in, I'm obviously out of time, in some way that we don't really understand. In principle, what one would like to do is then take these attention models and somehow, you know, look inside the black box and figure out what's going on. We've tried to do that in various ways, and it doesn't really make sense to us. Like, it's not interpretable in some obvious way. But I mean, maybe we just need to look harder. All right, I'm out of time. I'll stop there. This is just a reminder of who actually did the work. Yep.
Yeah, maybe we'll take 2 quick questions while the next speaker sets up.
No, no, the things are, they can be negative. You can have a negative effect on the phenotype. So because the 0 is arbitrary there, these things can and will be negative. And it just means that allele tends to reduce the phenotype. Yeah. So I think we did try a variety of other kinds of approaches. This attention mechanism has worked best out of anything that we've tried. I'm not sure, but basically trial and error, I think, is the bottom line. Is that reasonably accurate, Gautam? Oh, yeah, that's true. That's a little bit of a different class, yeah. But we did try a variety of different architectures. Yeah. Yeah. So, I mean, I think this gets at a bigger picture question. One of the things I think that's really going to be important is, you know, we're looking at these high dimensional things. We're trying to infer lower dimensional representations of them. We do so based on some set of environments that we've actually measured and some set of genotypes that we've actually looked at. And then, you know, a very useful thing to probe the validity of these models and their limits is to think about other classes of environments that you haven't measured, which would include things like combinations of environments that you did measure, and also other kinds of genotypes. So for example, one thing we're doing is we're taking these F1 offspring, we're back crossing them. It's basically like mating kids with their parents and different parents. And so you get different linkage structures, different relatedness patterns, and see how well things hold up. That's in progress, yeah.
Okay, so just in the interest of time, I'll stop questions for this talk. Let's thank Michael again for a great talk.